Hello, everyone. This is Martin Wiedmann, and we are ready to start today's webinar on Listeria environmental monitoring in the dairy industry. Quick introduction. I'm a professor in the Department of Food Science at Cornell University. The focus of my research and outreach programs is understanding transmission of pathogens, including and specifically focused on Listeria monocytogenes from primary production, whether that's produce fields or farms, all the way to finished product and consumers and everything in between. And as part of this project, this program, we have considerable experience working on Listeria environmental monitoring, not just in the dairy industry, but across different industries. And today I will bring this webinar to you. It should take about an hour. And after that, we will open it up for question and answers. You can type in questions in the boxes at various times while we're on here, and we will manage them. And if something is specifically pertinent, we might answer the questions as they come up. With that, here are the take-home messages for today's webinar. So I think what I want you to take home is that the processing plant environment is a very important source of foodborne pathogens, and that environmental sources in processing are particularly important for Listeria monocytogenes. We're going to use that information to show you how to set up a good pathogen environmental monitoring program and to emphasize that they need to be set up to find pathogens. That is one key learning, and the importance of this will underline by showing that if it is there, if Listeria is there and you don't find it, ultimately there's a high risk of regulatory agencies finding it, and the consequences of this will be much more severe. We'll also talk about increasing trend overall to require environmental monitoring, including through FSMA. And then we're going to have a major emphasis on really how to use environmental monitoring programs to drive corrective actions, because sampling itself, testing itself is not going to take care of a problem, but what you do with the testing results will. So that's going to be a major focus of this webinar. What I'm quickly going to do here now is I'm just going to change my shared screen because you're seeing a split screen, and hopefully this will be better now. Okay, so you should see the full screen now. Okay, with that, this is the outline of what I'm going to cover. I'm going to start with just sort of summarizing why we're so worried about environmental pathogen sources and why this concern about environmental pathogen sources is really more severe and more important now than it has truly ever been. And one driver of this trend that we see towards concerns about environmental pathogen sources has to do with the fact that we have new tools available to track foodborne pathogens to source. And these are tools that we call DNA fingerprinting. And I like this slide because it illustrates the concept. And so you can see here we have a number of Listeria monocytogenes isolates, but you can see that with DNA fingerprints, in this case it's just barcodes, we can say the first Listeria is different from the second one is different from the third one. Now this system is used US-wide and has been used US-wide to characterize all Listeria from human cases. So anytime there's a human that is diagnosed with listeriosis, if the Listeria monocytogenes is obtained, it is characterized by DNA fingerprinting as shown here and added to a database. Anytime an FDA, a USDA, or a state ag and markets department finds a Listeria monocytogenes in a food or even a food processing facility, that isolate will be fingerprinted and added to the same database. And that would help us to identify a couple of human cases that are caused by the same Listeria monocytogenes, even if they occur in different states, and say, well, we got two humans with the same Listeria subtype, they probably consumed the same food, launch an investigation, and we can trace it back. Sometimes, the more cases, the more likely to a specific food. And then fingerprint data for food isolates or isolates from environments of food processing plants can help in that analysis, because this sometimes might give us clues as to which facility might be a likely source and can confirm that a specific food is a source if we find the same isolate in a food as we find in two human cases with clear evidence that those humans consumed that given food. So that's really been a situation and a tool that's been around for basically 20 years now. Over the last 3 years, the picture has slightly changed from a technology side, where we moved from these banding pattern-based fingerprinting methods to fingerprinting methods that use the whole DNA sequence of bacterial isolates. So the genetic material of a bacterial isolate is made up of A, C, Ts, and Gs, as you can see here. And what we can see here is that we can characterize one isolate shown in top and another one, and we can say that these isolates are very similar. Most of the A's and C's are the same, but they're different in one. Here in the middle, you can see one is an A, the other one is a T. So in space, you can think of this as a better mousetrap. Very simplistically speaking, it allows us to do even better fingerprinting, more unambiguous to interpret, because we don't have to look and stare at banding patterns, but we have very clear letters that we can interpret very easily. This tool's been routinely used by CDC in the US and the FDA since September 2013 and had tremendous impact. And this is a picture that illustrates this. So what we see here is the number of listeriosis outbreaks detected every year in the yellow bars. So what you see from 1983 to 1997, we had one outbreak on average about every 3 years. So one in 83, one in 85, none for 3 years, then one in 89, none of them for quite a while, then one in 94, etc. Starting in 97, 98, we see more listeriosis outbreaks. Typically 2, some years 5, some years 4, etc. For an average about 2 to 3 outbreaks a year. What happened in 97? That's when we started to routinely use these banding pattern-based fingerprint methods. With middle of 2013, so once we look at the 2014 data, we now suddenly at about 8 outbreaks a year. What happened there? That's when we used DNA sequencing as a tool to detect listeriosis outbreaks. So we went from 1 outbreak detected every 3 years to 8 outbreaks per year. So 25 times more outbreaks detected. Now, food did not get less safe in the US over that time. Listeria did not become more of a problem. These outbreaks were always there, but we just started to detect them. As a matter of fact, if you look at the blue line here, that shows the total number of listeriosis cases. So many cases aren't outbreaks, it's just a person gets sick, and it's not 2 or more people linked to the same food. That number went down considerably, from about 8 cases per million people per year to about 2 to 3. That's about a 3 to 4-fold reduction of listeriosis cases. So food got about 3-4 times safer, but we have about 25 times more outbreaks, showing that the likelihood of an outbreak detected today is probably 80 to 100 times higher than it was 20 years ago. And that is why we see such an emphasis on listeria, because we're detecting and tracing back a lot of outbreaks that were not traced back before. Now, in addition to detecting the outbreak sources, these tools also really provide a much better understanding of the root cause of the problem. And then, to me, none illustrates that better than this outbreak, which was a big one that was identified in 1998-99, 108 listeriosis cases, 14 deaths, plus 4 miscarriages, linked to a processing facility that made deli meats, including frankfurters, very specific plant with fingerprinting linked back. This plant had a HACCP plan. It's a meat plant under USDA inspection. Had a HACCP plan. There were no concerns about the HACCP plan specifically. And before the outbreak, the critical control point, time-temperature combination, was under control. There was no issue there. But Listeria contaminated the product after the CCP. So basically, wasn't a problem with the HACCP plan per se, but it was a problem with the prerequisite programs. In this case, SSOPs, probably sanitation standard operating procedures, sanitary equipment design, et cetera, et cetera. So, and this is what we've really seen with all these outbreaks detected in virtually all cases. the root cause is not a problem with the CCP, but the problem is prerequisite programs and, in virtually all cases, contamination from the environment after the CCP. And this is why we realized that environmental monitoring programs are so important, because it is the one way to make sure we don't have listeriosis outbreaks that are linked to a specific facility. This illustrates what happened after whole genome sequencing. This is an outbreak linked to Karoon Dairy. 24 people linked to this facility. The second bullet really is the important one. In this case, FDA found listeria from 2 environmental samples that were collected in a manufacturing facility. When these environmental isolates were whole genome sequenced, so DNA fingerprinted, they found a number of isolates from humans that were already in the database that matched these environmental isolates. So we find listeria in the environment, and now we ask the question, are there any humans that were infected with this listeria? And in this case, linked it back. So it makes outbreak investigations much quicker, much easier, because rather than having 2 people that got sick with the same listeria, us trying to find out what food they ate, we know there's a listeria in a specific facility, and we ask, are there any people that got sick with it? The other thing that illustrates is the risk of having listeria in the environment. If FDA comes in a facility, finds the listeria, they will fingerprint it and add it to the database, and in some cases, then that will allow them to link an outbreak back to the listeria in that facility. If you have a good environmental sampling program, you shouldn't have listeria in your environment. If you don't have listeria in the environment, it doesn't cause an outbreak, and FDA can't find it, or USDA can't find it, and they can't add it to the database. So even more important today than ever to have programs in place to monitor the environment and identify and eliminate listeria from the processing plant environment. The latest outbreak, which triggered us doing this webinar, was a multi-state outbreak of listeriosis linked to a soft raw milk cheese made by Walter Creamery, a New York creamery. The details are shown here. It was a nationwide distributed raw milk cheese. At this point, the outbreak is considered over, but it affected 8 people in 4 states, including 1 person who died and 1 illness in a newborn. The evidence, in my opinion, and I'm going to editorialize this a little bit now, is the evidence that this cheese was responsible for the outbreak is very good. To identify the same isolate or close related isolates by whole genome sequencing from the cheese and the patients. The challenge in this case is that it was a raw milk cheese, and so there is a possibility that listeria could be in the raw milk, survive cheese making, and then subsequently grow, or it could contaminate the product from the environment. In this case, the investigations only focused on the processing plant. There were no isolates from the processing facility, so we did not have evidence by fingerprinting that the environment was responsible for this outbreak. And to the best of my knowledge, the raw milk source was never investigated. So therefore, we cannot exclude, in my opinion, the raw milk as a source, even though it is portrayed as it being an outbreak definitive link to the environment of this facility. I have not seen any data out there that would support that. To me, I don't think the investigation has gone into as much depth as it needed to assess that. I think that's an important thing to talk about. Unfortunately, there's not much we can do about it at this point. But we should not take this as an example that shows that environmental monitoring doesn't need to be done. It needs to be done. It needs to be a stringent plan to make sure that the risk of it coming from the environment is reduced to the greatest extent possible. And I'm happy to entertain at the end any other questions about this outbreak. Now, these same tools that have been used to identify disease outbreaks have also been used to effectively and better understand how listeria gets on a product. The example I'm going to give you here comes from a seafood plant, but I'm going to bring this back because we've seen exactly the same picture in dairy plants, whether it's cheese plants or other facilities. And so this is an actual study where we went into a facility This is one facility I visited 5 times. I'm just going to show you 3 visits. Every time we visited the facility, we collected samples. When we found Listeria monocytogenes, we fingerprinted it. And you can see the fingerprint both as a name, like 1039C in the first row, where the sample came from, and then the actual pattern. So we found Listeria on the first visit. Second visit, we found Listeria in a number of different samples, from floor drains to finished product. And third visit, again, we found Listeria in a number of samples, floor drains, finished product, product in processing. The key thing here is that we found the same fingerprint of Listeria on the first visit in a floor drain, and this one is named 1039C and marked with a star. The second time in multiple samples, from floor drains to fish that were being brined to finished product, and again in the third visit, mainly in floor drains, but again in fish being brined and finished product. And the fish we found it on ranged from fish where the raw material came from Alaska to fish where the raw material came from Norway, Faroe Islands, etc. So a variety of different raw materials, but all ended up contaminated with the same fingerprint of Listeria monocytogenes. These visits occurred about 5-6 weeks apart, so we could find the same Listeria in this plant over 18 weeks. Conclusion we drew from that is that most likely this Listeria lives, persists in this facility over time, and contaminates product that comes in from different sources. And we have some very good evidence supporting this DNA fingerprinting data that are shown here. So it's sort of suggesting and supporting the importance of environmental sources for Listeria contamination of finished product. Now, this study was somewhat unique in that we actually looked through 3 facilities that were all in close proximity of each other, about 15 miles, all making similar products. The facility I just showed you is facility D, where we found fingerprint 1039C in 10% of samples. We never found that Listeria in plant C or plant B. So it seems to be specifically living in plant D. If we look at plant C, this fingerprint of Listeria 1046B, marked in red, only found in that facility, never found in facility B, never found in facility D. So different type of Listeria, but that one seems to survive in this plant over time. So 2 different facilities, each of them has a Listeria that is unique to it, and this is what we sort of call pet Listeria. Pet in animals, little animals, which bacteria are to a certain extent, that live in your facility over time. Plant B has 2 of these pet Listeria that survive in the plant facility, 1042C, 1045, both of them we found basically every time we visited that facility. So it shows that many plants, particularly if there's no stringent management and environmental monitoring program, can and will have Listeria that survive over time. And the data that I'm not going to detail is that very often these Listeria are the ones that then are most likely to go on finished product and cause problems. Next slide illustrates the complexity and the challenges a little bit more. Again, another facility we worked with. What we have here is from top to bottom, basically the flow of the product. We have raw material come in, then we have the raw area where the materials is staged to be ready to be smoked, ready to be processed. We have the finished product area where products are being sliced, packaged, etc., and then we have finished product. Every column is a month, and that means we visited this plant monthly over 12 months, collected samples, and tested them for Listeria monocytogenes. And I'm only going to focus on the Listeria monocytogenes data here. If we find Listeria monocytogenes, we fingerprinted it, and we color-coded these Listeria monocytogenes fingerprints to make it easier to look at. And so what you can see here is that you have a number of different colors, so a number of different Listeria fingerprints, but one of them more common than the others, and it's the fingerprint that's shown in green and is named 1053A. And we find that fingerprint of Listeria over the whole year, but not every time we visit the facility. Find it in a drain on the first visit, don't find it for a couple, find it in an apron of an employee, etc. Once we find it on a sliding door in the finished product room, Other than that, we only find it in the raw area. The apron of the employee, where it shows up 4 times, is a single-use apron. So clearly not the place where this listeria would survive. It's a single-use apron. But this is an employee that handled pallets that he picked up from the floor, etc. So could be likely transfer from the environment onto the apron. So based on this, we concluded that this plant indeed has a pet listeria, that it probably lives somewhere in the raw area, but we don't sample the right places yet to find exactly where it lives. But in that plant, there are issues, and sometimes this listeria can be moved from the raw area to the finished product area, and we hypothesize that it's most likely by some sort of traffic, either equipment or people, based on the fact that we're finding our sliding door handle, most likely people. So it suggests that we have some problems with separating raw from finished with hygienic zoning. So our environmental sampling plan in this case shows us listeria, persistent listeria problem in the raw area, some issues with zoning. Now, for the next 12 months, we did in-depth sampling in the raw area to identify where this problem came from. Lo and behold, when we took samples of other areas, we had one site where we found the green listeria every time we sampled. And that's shown here. It's a composite sample of multiple floor mats. What does this look like? This is the type of floor mat, not the exact floor mat that we saw there. It's about probably 1.5 inches, maybe 2 inches thick. Most likely what we could conclude is that listeria sometimes was embedded in the interior of this floor mat. If these floor mats were foamed with sanitizer foamer, the sanitizer would not penetrate to the center. Even if they were submerged in a sanitizer solution, the sanitizer would not penetrate all the way to the middle. So the listeria could protect themselves, could hide from sanitizer, so to speak, in these floor mats. But when the floor mats were put back on the floor and people stood on them, then that weight of the people would propel or move the listeria back out of the floor mat and put it back into the plant environment. Therefore, effectively, these floor mats served as a niche, a protective site where listeria can survive sanitation. When we removed these floor mats, we never saw that green listeria subtype in the plant again for at least 6 months. Now, year 2 also gave us another couple important learnings, and one of them is that we continue to have hygienic zoning issues. Listeria was found again in the finished product area, and even moved to food contact surfaces. On the bottom right, you can see that green subtype showing up twice. One is on a slicer, one is a finding on the hands of a person handling product. Maybe even more striking and important is that we've never found this green listeria in the finished product, the very last row, even though we collected 6 samples every time. So this shows that a good environmental sampling program can detect a problem, identify the root cause of a problem, can help us to eliminate that before we actually find the listeria in the finished product. Now I'm saying find, not before it makes it into the finished product. There's a high likelihood that it made it onto the finished product, but just at such infrequent intervals that sampling of 6 products was not sufficient to detect it. So environmental modeling is a very, very good, very powerful early warning system to allow processing facility to detect problems before they show up in the finished product. What I've shown you there with the floor mat is the idea of a gross niche. Now, this is not just floor mats. This is a concept that's been known for a while, and another textbook example of a gross niche, so a location that can harbor listeria after routine sanitation, is our hollow rollers as displayed here. So you can see the crack between the metal roller and the plastic here. That small crack would allow water, some dirt, and potential listeria to move inside the roller. Once inside the roller, it's protected from sanitation because the sanitizer doesn't effectively penetrate. But if that roller rolls for 8 hours, there's moisture, some of the listeria might migrate back out, contaminate the roller, contaminate the conveyor belt, and then contaminate product on the conveyor belt. So another area that could be effectively served as a niche for Listeria monocytogenes. And if that wasn't enough to convince you, here's what the inside of a roller would look like. So you can see these dirt accumulates, residues accumulate, and it's a likely or possible source or location for bacteria to survive. This is not just survival of Listeria, but this sort of idea of Listeria surviving a processing plant and leading to disease outbreaks is also very well documented. Here's a dairy example, actually an outbreak in Finland, where Listeria seemed to have survived in this facility for at least 2 years, caused 25 cases, 6 deaths due to butter, not a product we typically hear about with regard to Listeriosis outbreaks. And the most likely source was described as the screw conveyor in the butter wagon. Now, I wasn't in that facility, but it is very easy to envision how a screw conveyor in a butter wagon might not be the most easy thing to effectively clean, and how this location could have harborage sites that allow Listeria to survive over time. How challenging can it get? And this is an example of an outbreak in the US. In 1988, in this facility, one Listeriosis case was linked to hot dogs produced by this facility. 12 years later, different owner of the company, different products produced, the same product from producing the same physical structure, the same facility, was linked to 29 Listeriosis cases linked to consumption of sliced turkey meats. The Listeria monocytogenes stayed the same. Top two Listeria fingerprints are Listeria from 1988, human and food. The rest of them are Listeria from 2000. So this is a pet Listeria, very old pet Listeria that survived this facility probably for 12 years. And this is not the only example of this. So it shows that Listeria can, particularly in older facilities, survive for quite prolonged times and can continuously or in intervals contaminate food and cause problems. So further illustrating how important it is to identify spots in the environment where these organisms survive and eliminate them, which is what we call seek and destroy. What I've shown you in the last few slides really over and over again is the importance of that methodology, but also that attitude of seek and destroy. You go into your environment, you seek Listeria monocytogen, and then you destroy the place where these pet Listeria live so that they cannot live in a facility over time. So with this sort of introduction to how does Listeria get onto the product, what is the root cause of the problem, I hope I've convinced you that persistent environmental contamination with Listeria is important. It's the root cause of many problems. And it has been reported in almost all types of food processing plants, from seafood to dairy to meat to produce, you name it. These persistent Listeria contamination issues don't just cause problems as you have the same Listeria in the facility, but it causes repeat finished product contamination, and that then can and does lead to Listeriosis outbreaks. And virtually all Listeriosis outbreaks recorded have been linked to persistent Listeria contamination. One way to address this is seek and destroy, which then, of course, moves us into environmental monitoring programs. Because seek and destroy, the idea is linked to environmental monitoring programs. So while we're talking about Listeria monocytogenes in this webinar, I just want a short reminder that that is not necessarily the only pathogen we target. It is the main pathogen. So key pathogens that we might target in environmental monitoring programs, Listeria monocytogenes, we talked about it. One of the challenges here, and this is important, throughout the food industry, is that Listeria monocytogenes is actually a common organism. We've done studies in New York State and found Listeria monocytogenes in 7 to 8% of samples we collected from urban environments. And I'm talking about going out to Syracuse, New York City, and swabbing playgrounds, sidewalks, taking water samples from rivers and ponds. On farms, sometimes we found 20 to 30% of samples being positive for Listeria monocytogenes. What does that mean? Listeria is going to come into your facility, okay? Can't tell you how often, but it will come in. So you will find occasional positive for Listeria monocytogenes. What you should not have is a place for these Listeria to hide in your facility where now they can survive over time. Your monitoring program should monitor your procedures that minimize listeria coming in through procedures such as changing shoes, et cetera, but also, and very importantly, should monitor your procedures to make sure it does not and it cannot survive in a processing facility. Salmonella is the number 2 target for environmental monitoring programs. More of an issue in dry processing facilities, cereal, bakery products, dry powder, dairy powders, etc. And then the number 3 target, which is really only in a small subset of industries, Chronobacter, which is a challenge and a concern in infant formula manufacturing facilities, so probably not applied to many of you or any of you on this call. So now let's get into the bulk of how we design and implement environmental sampling programs. I will point out a few things in this new guidance from FDA that just came out that some of you may have heard, but I strongly encourage you to take a look at it. So this is a guidance that the CFSANS, or the Center for Food Safety and Applied Nutrition from FDA, published in January 2017. It really lays down the current thinking of FDA with regard to environmental sampling. And there's some significant differences from how FDA approached environmental sampling before, including making some very clear statement that there's an expectation for companies to sample food contact surfaces. But this is a draft guidance, so we will see comments on this, and it might not be the final document, but still. So much more of an expectation of sampling food contact surfaces. Also very clearly lays out that environmental monitoring for listeria should happen on samples that are collected at least 4 hours into production. So for those plants that have done traditionally what we call pre-op sampling, so you clean, you sanitize, etc., you sample, and then you start, that's not going to cut it anymore. We really need to sample at least 4 hours into processing, which is actually consistent with some of the regulations in Canada and some other countries, but is going to much more likely yield positive results. There also is an expectation of sampling food contact surfaces, but also some more clear language that aligns FDA guidance with USDA guidance now, and makes it clear that just because one finds listeria species, not a mono, listeria species on a food contact surface, does not mean an automatic recall of their product. And I'm not going to get into the details of that because that's another hour by itself, but the details are found in this draft guidance. If you have not looked at it yet and you do listeria sampling and you are an FDA-regulated facility, strongly encourage you to read this. We can't talk about environmental monitoring without talking about FSMA. FSMA laid down the law pretty clear that environmental monitoring is considered a required program. It is not a preventive control, but it is a strategy, a tool that verifies the effectiveness of sanitation programs. So it verifies one of your preventive controls, which is SSOPs and sanitation programs. It also is there to verify hygienic zoning is working. And giving you some examples of how that would work with the graph and the pictures of the environmental sampling we did where we color-coded the listeria. Additional sort of goals of an environmental monitoring program as detailed in FSMA is to understand normal environmental conditions, so we can see when we're in an enhanced risk situation because we find more positives, and it clarifies that it's not a preventive control. Key for as we implement this is that an environmental monitoring program must be tailored to each facility. It's not one size fits all. It may include pathogen testing as well as testing for indicator organisms. So indicator organisms could be Enterobacteriaceae, could be total bacterial counts, etc. And these documents also very clearly outline, and the FSMA training very clearly outlines that a good environmental monitoring program must try to find the organism. And we will get back to that. Another way to sort of think about environmental monitoring program is really shown here, which is sort of called the listeria control equation. And there's two similar equations. I'm going to focus on the one at the bottom first. It basically shows that there's strategies in place in every food processing plant, including dairy, even though it was specifically designed for dairy, that control listeria. And these key strategies are separating raw from ready to eat, so hygienic zoning, in other words, GMPs, sanitary facility and equipment design, effective cleaning and sanitation procedures. That's what every facility has to do and will do. Our environmental pathogen monitoring program is there to make sure to verify these facilities and make sure they are working effectively as intended. If you combine all of that, that's when we have an effective pathogen control program. Now, the same program will also control environmental spoilage organisms. It's the same niches that allow Listeria to survive over time. The same problems with limited separation from raw to ready to eat also move spoilage organisms around and cause issues there. The top graph basically says the same thing, but focuses on some more of the more specifics. So I'm saying separate raw from ready to eat. It talks about controlled traffic patterns rather than, and it provides some specific examples of environmental controls, such as clean, dry, and uncracked floors. And this is an important one, is that experience and data very clearly show that if you can keep a plant dry, you reduce the risk of Listeria contamination and survival tremendously. So drying out plants, keeping plants dry, very, very important strategy as we control Listeria. And a related concept and mindset is, of course, seek and destroy. I'm not going to talk through it too much, but this is just a slide that illustrates it, and also a link or provides sort of a reference on something that we published on Seek and Destroy. At this point, we're going to make these, both the presentation will be recorded, we'll make that available on our website. We'll also make the slides available so you can use them for internal training, etc., and can download them. So let's talk about the nuts and bolts now. Let's talk about designing environmental sampling plans. So sampling plans need to be developed individually for each plant. We talked about it as a trend. There also is a trend towards regulatory agencies not just recommending environmental sampling or requiring it more or less, where we're going to be very soon, where we are with FSMA, but also that regulators will do environmental sampling. Many, if not all of you, have heard of the FDA swabathons, where FDA will come in and may come in and collect 100, 150 samples from one facility. That could be due to specific assignments to assess risks with a specific type of product, or could be in response to a review of a facility which does not look like that facility had a robust enough environmental sampling program. Again, the idea here is that you as a processor, you as a facility have an environmental monitoring program that makes sure you find Listeria before an FDA comes in and before they might find it. Where do we test? We can look at test sites in terms of food contact surfaces and non-food contact surfaces. Again, go back to the FDA guidance document or USDA guidance document to see what you have to do if you find a positive on a food contact surface. Still a very important issue to consider, because you might move into a situation where you have to do test and hold, or have to do finished product testing. Not necessarily with the first positive samples, but definitely with subsequent ones. The more common way to look at where to test is to look at zones and to separate a plant into zones. And this slide illustrates it maybe a little bit more easy. Most plants will have zones 1 to zone 4, with zone 1 being food contact surface, zone 2 being non-product surfaces that are near those contact surfaces. So that could be framework of equipment, could be switches, could be control panels, etc. Zone 3 are other areas within the finished product room, and then zone 4 are areas outside of the ready-to-eat room. For FDA plants, before and this new guidance came out, I would say typically 90 to 95% of sampling focused on zones 2 and 3, maybe 30-40% zone 2, 50-60 of zone 3, and typically we see 5 to 10% of samples taken in zone 4. That's not a law or a rule, but it gives you some guidance what people in the industry generally do. In some facilities, depending on, there'll be more zone 1 sampling. I'm a strong believer that some zone 1 sampling should be part of each environmental monitoring program, because we have seen situations where you can have a niche in a zone 1 that you might not necessarily detect by sampling other areas. Where to test, the other way to think about it is whether we sample actual niches. We talked about them, provide some other examples here. Whether we sample transfer points, these are areas that move Listeria around. So sampling transfer points can help you identify particularly issues with your zoning, with your hygienic zoning, with your raw finished product separation, etc. And then some areas can be both. These conceptually think about a computer keyboard that is touched by hands. If I sample that 4 hours into processing, now the Listeria could survive on the keyboard, particularly since I typically don't sanitize it that much or that easily, but it could also be a transfer point because people touch it with their hands. Now, the key part of all of this is we can develop the perfect sites, find the perfect sites, but we need to collect the samples so that we find positives. Unlike finished product testing, the subtleties of sampling are much more important with environmental sampling than finished product samples. And I'm going to illustrate that with the next slide. And this is a situation you might see in a number of facilities that you have an environmental site, a floor like this, and your sampling plan might call for a sample floor at filler 3. And this is what the floor looks like. Now, here you have options. One option is to sample on the diamond board, the metal surface here. You could sample on the floor tiles, or you could take a sponge and squeeze it in the gap between the two of them. If you want to find Listeria, you probably squeeze it in the gap between those two. What I do, I actually stand on the diamond board because it's loose. If you stand on it that way, put some of the water moisture, comes out in between those two, makes it even more likely to find Listeria. On the other hand, if I don't want to find Listeria, if I just want to have nice, clean records to show regulatory agencies or my customers, I'm just going to collect a sample on top of the clean diamond board. Okay? Tiles are somewhere in between. If I have a loose tile, and this illustrates another sample of some of these. If I have 50-some tiles here, one of them is loose, I need to sample around the loose tile because that's where I'm going to find it. I sample 2 tiles over that are well sealed, no cracks in the seal, much lower likelihood. So the specificities of where I sample, where I stand, how much pressure applied to a sponge make a big difference in terms of whether I find an issue that's there or if I don't find it and fool myself into thinking I don't have an issue. The problem then arises if we have policies and messages that actually don't encourage people to collect samples. And I'm going to only pick one of these as the middle one, and I'm going to take it a little bit further. If a plant has a KPI, a key performance indicator, it says we want less than 1%. Our goal is less than 1% Listeria environmental positives. And if a plant reaches that over a year, they get a bonus. In that situation, do you think people will sample this crack if it's end of year and they've been at 0.8, 0.9%, and a couple positives would push them over the 1% mark, and therefore the whole plant will not get a bonus? So KPIs, great idea for productivity, and some measures will not work with with environmental sampling. And other examples here that you can sort of read through on yourself. So the important thing is need to set up a systems, need to set up messaging that makes sure people actually correct the samples that will find positives. And here are a few other examples. Yeah, obviously here you would want to sample the cracks in the wall, not the clear tiles. Obviously here you want to squeeze a sponge in under that joint, under here, not just sample a clear surface here. Under here you want to sample that wall to the right, to the bottom, which seems to have much more cracks between the floor and the wall, not the wall on the left where you have a good, reasonably well-designed floor-wall junctions. So back to those conclusions, and to summarize it, you know, I hope these things illustrate to you that it's not trivial to encourage people and set up a system in your plan where people will collect the right samples, but it is absolutely essential. When to test? Obviously, this can occur pre-op, mid-op. Pre-op in many facilities still done. So that's after cleaning and sanitation before operation starts. Easy to interpret. If you have a positive in a spot there, probably means Listeria survived, but much less likely to yield positive samples. We've still seen facilities that think they know they have no problem because they take a few pre-op samples that have always been negative. The picture could be a very different one if you collect samples 4 hours into processing or later. Increasing guidance is sample at least 4 hours in, more likely to yield positives. It really helps you verify your food safety plan and your strategies. And they'll give you information on hygienic zoning, but will require follow-up, because the sample size that is positive does not mean that's where the listeria survives. And again, I may want you to think back to the green listeria. Where did we pick it up? On the apron. That wasn't where it survived, but we sampled mid-op. We had to do follow-up sampling, what we call vector swabbing, where we take samples around the positive area to find the actual spot where it survives. If you do vector swabbing, when you do root cause analysis after an initial positive, then you might collect samples mid-op and pre-op. And the pre-op might then tell you where it really survives. But for your routine monitoring, you want to sample at least 4 hours in. And in many cases, you have to. When to test? Again, there's a gold standard of what people do. The gold standard is a random number generator. What do I mean with that is, we say for next month, we're going to randomly pick a day, and we let a computer program randomly pick the day and the time after those 4 hours when we sample. Reality in most plants is we sample maybe Monday through Wednesday, because we don't want to have the lab have to work over the weekend. The lab prefers the samples earlier. And then we sample when it's not very busy in the plant, because that's when QA has time. Problem with sampling when it's not busy is that that's also when you're less likely to have listeria issues. The more busy a plant, the more shortcuts are taken, the more likely you have a listeria issue. You want to sample, or at least have a good chance of collecting samples on days when the plant is running over time and running very busy. In a perfect environmental sampling plan, every day of the week has a similar likelihood of being sampled. How to collect samples? There are different options. There's sponges. Some people talk about wipes. There's good swab, like you see on the right side here. For pathogen monitoring, we recommend sponges. They can sample bigger areas, sample them more effectively, you're more likely to find pathogens. Swabs would only be limited to very, very specific situations where you try to reach difficult to reach areas. If I go into a facility, collect 100 samples, 98 to 99 will be sponges, 1 or 2 might be swabs. We look at the picture on the right when this person samples with a Q-tip sponge swab-like thing. You see a number of issues. First of all, we have what people call a template in order to sample a defined surface area. And it's sample a nice, clean, flat surface, right? But if you look at this picture, where should we really sample? We should sample in the crack that is shown on the left side of the sampling area, maybe in some of the corners and edges here. So the visual image that people still have, even if they don't use a physical template, is that you're supposed to sample a defined area. And some instructions even say that sample foot by foot. That's not what we recommend. We recommend that you sample difficult to reach areas, which typically cannot be measured in square feet, because we might just sample 2 yards of a crack between a floor and a wall. Very different. So no templates, no specific areas for pathogen sampling. If I do an ATP swab, I might use a specific area to have comparison. But for pathogen sampling, no. For pathogen sampling, we focus on sponges and difficult to clean areas. How often do we test? That can range from every day, multiple times a day, weekly, and then some very small operations might even be monthly. Most cases, weekly would be a minimum. If in many situations where we have more sites than we have budget or time, we might have a large set of sites, let's say 200 potential sites or 30 potential sites, and only a subset is sampled every time. And that subset, again, is selected randomly. We might have 2 sets of sites. We might have sites that have to be sampled every time. I have 100 sites. I have 10 high priority sites that are sampled every time. The other 90, we only pick a subset, like 30 or so. Questions we often get is, how many samples should we really collect? And there's no guidance, really. There's no rule. There's no regulation. But for those who really want some very specific guidance, here is some. and this is from the Innovation Center for U.S. Dairy, really one of the few people that put out a guidance. And this is really only be thought as a rough area. So it basically goes minimum 30 swabs per 50,000 square feet per week, best of class, more than 55. So rough rule of thumb could be 50 per 50,000 square feet, so that's 1 per 1,000 square feet per week. If you're way below that, you probably have to rethink it. If you're in that ballpark, you're probably doing a good job, but it also needs to be adjusted. So older facilities with past problems, you would typically collect more samples than that. On the high side, if you have a facility that has low pathogen prevalence, a good plan, a long history if it's new, and you may have moved from some pathogen testing to indicator testing like total plate counts, etc., you might have fewer. Test methods, you have the whole range available from the traditional methods to DNA-based methods. Traditional methods are often time-consuming. DNA-based methods should give you results in a couple of days, so it helps you to do a root cause analysis much more quickly, much more effectively, because if you get a result 6 days after you took a sample, very often it's hard to go back and figure out what went wrong. So I'm a fan of DNA-based methods just because of the improved specificity. You're less likely to get false negative results when the test says negative and it's very positive if it's a good test. And yeah, that's something we could discuss if needed. But really anything goes if it fits within your overall scheme. What to do with testing results. So now we're moving into, okay, we do testing, but are we really going to make food safer? Are we really going to reduce our business risk? Is through what we do with the testing results. Results should be regularly reviewed, including every time the results are reported, but you shouldn't just look at the results you get that day from your lab. You should look back. You should try to get at least an idea of medium to long-term trends. Results should be organized in one specific location, and that specific location, this is very important, should also have the documentation of the corrections you took if you had a positive. There's nothing more damning in an audit than you having a record of a problem, a Listeria environmental positives, and you not being able to find the record of the corrective action. Put them together, have them in the same spot so that does not happen. You should also have a policy and a plan in place to do regular, more in-depth reviews, whether that's every 3 months, every 6 months, every year, depends on thing, but there should be a plan, a proactive plan to do this and implement this regular overall reviews. One thing that we find very good strategy is to also have a floor plan, so you can map out your sites, as you see here. So these little circles are our sites. You can see they're numbered, and then there's a detailed legend that tells us what the site is. And we have little boxes here. If we have a colored box, it means it was positive, different colors, different fingerprints. Most people won't do fingerprints, but at the very least, you can get a visual in terms of where in your plant do you see positives. Okay. We're going to take a little bit of a detour here for 3 slides, which is sort of like, okay, I have a sampling program, I'm running it, I'm looking at my results, I'm analyzing this, but a lot rides on this program. How do I know I have a good program? What I'm going to give you as an example of 7 plants we worked with, and we helped them develop the environmental monitoring programs, and they implemented them, they ran them, and this are the results we got from them cumulatively. And you can see if you look at these plants and look at the percent Listeria positives, it looks like a wide range of plants. The plants like plant F, that seems to be a very good plant, has Listeria under control. No Listeria positives out of 334 samples. Down to plant H, which some might say, wow, 23% of samples positive. That's a high frequency. Okay. And your plant could be any of those, but how confident are you that it really works? So what we did is we actually sent an independent team into every one of these plants. They collected a considerable number of samples, 50 to 100 samples, to validate or check this sampling plan. So here's what we found. So these are the same data in the first column that I showed you before. Here's what an independent group found. So for example, let's look at our 23% plant. When we went in as an independent group, we only found 4% of samples positive. On the other hand, if you look at our 0.3% no positive plant, when we collected 50 samples, 3 of them were positive. It's about 6%. So I color-coded these plants now in green, where we actually, the independent sampling found less Listeria than the actual plant themselves found, which to me is what I want. It's almost more important than the absolute prevalence. That means the plant is searching, it's finding it, it's taking action. Because of that, when we go and we find less. On the other hand, we have the red plants that found very low Listeria, but when we went in, we found considerably higher numbers. So these are plants that may not sample as stringently as they should. And then there are some where what we find and what they find match reasonably well. Obviously, your plant should be one of the green plants, but what I want to impress on you is that you might want to think about whether you do a regular validation, a check on your environmental monitoring program. And we at Cornell and our dairy extension team do this. We offer this for some plants. So if you're interested in that, you can do it internally to your company, or you can talk to us and we can help you set that up. So far, we've talked about pathogen monitoring. Now, there are other environmental testing programs, testing strategies there that can also help you and will help you to sort of make sure you have your environment under control to minimize the risk of contamination. One that is commonly used and is actually sort of more or less the standard strategy for controlling Listeria monocytogenes is what's called an index organism testing. So everything I presented you really was Listeria or Listeria monocytogenes testing. Listeria monocytogenes is the organism that causes disease. Listeria species is an index organism. So that's defined as an organism that, if found, relates to the possible presence of a similar pathogen. What does that mean in common words? If I find Listeria species, that means there are conditions in my facility in that specific location that would allow Listeria monocytogenes to survive and thrive. Therefore, I need to fix it. So, Listeria species, similar pathogen, Listeria monocytogenes could be there. Indicator organism does not tell me something about a specific pathogen, but it tells me something about the microbiological and the hygienic condition of a food, or more importantly, in this for us here, an environment. Typical indicator organisms will be Enterobacteriaceae or standard plate counts. Classically coliforms, but really data accumulated over a long time have shown us that coliforms is not really that good an indicator. Very often we find coliform negatives and we can still have considerable problems. We can also test for spoilage organisms. We can test for ATP. ATP really tests for presence of organic material. And why is often be seen as a sanitation monitoring system? It really doesn't monitor sanitation. It monitors cleaning. It determines the cleanliness of a surface. Sanitation would effectively eliminate pathogens and bacteria from a surface, but an ATP test can be negative even if we have 500 to 1,000 bacteria. But if a surface is not clean, an ATP test will give us a positive. So an overall comprehensive environmental program will include tests other than pathogens and index tests. All right, so we have our program. How do we set up guidelines for follow-up and corrections? How do we make sure this program prevents contamination? These are some of the key guidelines, and we're going to cover a little bit more details on some of those. The corrections you take based on a positive plant, again, might differ by plant, by what product may you produce, and will differ by zone. A zone 1 positive will trigger a different type of action than a zone 4 positive. It's not just single positive that trigger actions, but a trend towards more frequent detection also needs to trigger action to deal with this. The typical initial 2 actions really after positive is to take more samples to do vector swabbing to see whether this contamination has spread and or to identify specific other areas where the organisms might survive, and to do what's called deep cleaning. And so deep cleaning will be just clean the specific area where it's found and the areas around. But these corrective actions must go beyond vector swabs and deep cleaning. We really need to identify the root cause. The worst case scenario, which I've seen in some facilities, that a facility has a positive, take 4 or 5 vector swabs around the area, pour as much sanitizer as they can find on all of these areas, resample 3 times right on top of the sanitizer. I have 3 negatives and say, we had an issue, we did vector swabbing, deep cleaning, and the problem is resolved. That's not a corrective action. That's not a root cause analysis. Good environmental monitoring programs and follow-up programs would drive corrective actions such as these. Changing cleaning and sanitation procedures and SSOPs. We recognize that we're not cleaning, our routine cleaning is not sufficient, that we're not taking apart equipment enough to really get at the niches. A good program will drive changes in maintenance, where we fix pieces of equipment to prevent niches, and drive changes in preventive maintenance programs. That we use environmental monitoring programs to tell us how often we have to change a floor mat, how often we have to change gaskets, etc. It may go to a point where we have drive that equipment be modified and replaced. One thing it's not on that list, but also very important, it may drive and should drive our master sanitation schedule. How often do we clean and sanitize equipment that we don't clean every day after every shift, but once a week, once every year, once every 6 months? Those timeframes may have to be modified based on these environmental monitoring programs. So this is where the rubber hits the road is, what are we going to do with these results to improve our facility? We recommend that you have a flow chart for what you do for corrective actions. And when you go back to your routine sampling, I'm not necessarily saying that this specific flow chart is exactly what you should be doing, but it's illustrated the type of procedures you want to have in place. We also strongly recommend that you have a clear form that you use to record every corrective actions. I'm a big fan of electronic records, but I'm also realistic that that might not be feasible, particularly for small facilities. We go in and train small facilities. We try to give them a simple sheet like the one shown here, which allows them to record the corrective actions, to say when the sample was taken, which side was found positive, what it found positive for, when the action was taken, and a description of it, so that you have at least some record to show in case of an audit. The other thing that is really important is to have actual procedures for root cause analysis. Some of your small processes that might seem like overkill, but what we actually do is we try to encourage people for every positive to complete at least part of a fishbone diagram, where you say, effect is our Listeria positive. What are the root causes with regard to people, equipment, culture, and process? So equipment could be root causes. Equipment is difficult to clean. Why is the equipment difficult to clean? Because it's old and we haven't repaired it. Because we're not doing preventive maintenance. Why are we not doing preventive maintenance enough? Because we don't have an actual schedule for preventive maintenance. And that then gets us to the root cause fix. People could be, why are we not sanitizing well? Because our sanitation team isn't well trained. It doesn't know what they're doing. Why they don't really know what they're doing? Because they're not trained. Why they're not being trained? Because we don't even have a training program, etc. All of these are fixable things, okay? And all the other things that can and should be discovered, facilitated by these environmental monitoring programs. Some specific examples. So this will be the type of environment you would sample. This is an underside of a conveyor belt. You have metal plastic interfaces here. There's a small crack. If you swab around here, you might find a positive. You might realize that Listeria could survive in that gap, in that tight gap between the plastic and the metal. And that then leads you to a change in procedure where you might say, every week or every month, we're going to have to disassemble these and do deep cleaning. If you say every month and you start to get positives after 2 weeks, then obviously environmental monitoring program tells you that the frequency was not correct and you fix that. But it's a way of how you can see how environmental monitoring can drive your master sanitation schedule. If you have areas like this, again, this might have to be taken apart regularly to allow better cleaning if you find positives here, because again, you have these tight metal on metal joints where Listeria and other pathogens can hide. Or you could redesign it, seal it, or make it more open and easier to clean. You can have areas like this, hollow roller, difficult to clean conveyor belt. You find this positive. Long-term, you want to redesign it like this. Short-term strategy, you might say that you have to remove your hollow rollers and submerge them into a clean out of place tank, high temperature, so that you control it with temperature, because you cannot clean and sanitize inside the hollow roller. So there's long-term strategies and short-term strategies that can be used to deal with these sort of issues. If you find areas like this, take care of the positive, poor welds, etc., your control is to fix these, get nice clean welds that now are easy to clean. But your root cause analysis also should ask, why do we have poor welds like this? Well, because our welder doesn't know how to do sanitary welds. Why do they know that? Because we never send them to training on sanitary welding. So that's another example of a root cause analysis that is done and completed properly. So we're back to the take-home messages where we started. I hope I've convinced you the processing environment is an important source of foodborne pathogens, particularly Listeria, and that because of changes in surveillance, regulation, and otherwise, environmental monitoring programs are more important than ever. Hope I've convinced you that a good monitoring program needs to find pathogens, and that's actually easy to have programs where, you know, we go into the motions, but we're not really doing everything we can to find Listeria. And finally, and maybe most importantly, I hope I've convinced you and shown you that it's really, really important to think about the actions we take after positive results, because sampling alone will not control hazards. It's what we do with the results, and that's really, really essential. Now, you might think we're done, but we're not. We have 2 more slides, leaving you with a few possible action items, you know, some tangible things. So if you're the company owner, it's one thing. If you report up, make sure the business risks due to food safety are known and communicated in your company. Remember the slides at the beginning where I told you that with today's surveillance systems and DNA fingerprinting, outbreaks are 80 to 100 times more likely to be identified. That's your business story right there. Make sure your company's leadership shows commitment to food safety and doesn't intentionally send wrong messages. You know, I gave you the KPI example. And then go back and assure that your company has a robust pathogen and microbial environmental monitoring program. Review the sampling plans, develop some strategy to validate your sampling plans. And again, very important, assure that your sampling results drive short-term and long-term changes, not just a little bit of vector swabbing and deep cleaning, and let's make sure we have some negative records. That is not going to protect you from some of these business and food safety risks. Do that when you come back to your plant, and if you're just starting a program, or if you have a program and you think it's ready to undergo some review, here's sort of some 10 steps that I recommend to start a Listeria monitoring program. Again, we will share these slides. You'll have them available to quickly run through. Assemble a team, conduct a risk assessment, and that sounds complex, but it's simply a literature search, understanding the environment to figure out what you should test for. Is it Listeria only? Is it Listeria and Salmonella? Get a floor plan of your plant, see whether you have some old monitoring data, put them on the floor plan to get at least some idea of what has happened in the past. Come up with your list of sampling sites, decide on when, where, and how you're going to sample. And then, very important, develop a flowchart of corrective actions. Do that before you start sampling. Come up with your record-keeping system before you start sampling. And only then start your sampling program. And then reevaluate it and validate it regularly. That's what I had. I want to thank my friend and colleague John Butts for some of the slides, particularly the ones that illustrated real-world situations. And with that, I'm going to open it up for questions. And let's see what we have here. So if you have questions, there's a webinar chat. So type in your questions there. That's the easiest way for me to see it and to respond to it. I'm going to give everyone a little bit of time to put in some questions here. So one question is, oops, there's another one came up. One question is, what do you think of the use of the 3M Petri film for Listeria as means of detecting Listeria species in-house? So I'm going to translate that question. So we talked about different testing methods, and typically environmental monitoring for pathogens involves taking samples and shipping them to a contract laboratory, or if you're a larger company, shipping it to your own testing lab that often might be located off-site. Now, there are some strategies out there that would allow you to make it more easier for you to do test environmental samples in-house. One of them is the 3M Petri film for Listeria. And for those of you familiar with the Petri film, it's a simple, easy way to test things. It doesn't require you to make these bacterial plates. The challenge with using this is, number one, is you grow pathogens in a lab that is close to your facility. That raises concerns because now you don't just have one Listeria, but when you grow Listeria on these Petri films, this one or few Listeria you have becomes typically in the range of billions of Listeria. And so if you have that many in your lab, what's the risk of reintroducing that or introducing even higher levels into your facility? So one concern is, do you really want to do pathogen testing in your facility? I would only recommend that, whether it's the Petri film or something else, if you have a reasonably sophisticated staff, people who have microbiology experience, microbiology training, separated facility where you can do it, very, very good separation to prevent reintroduction of these organisms and contamination of your facility. Typically for a lot of facilities that will be out of reach. The selling point for some of these methods, such as the 3M Petri film and some other ones, is that you don't grow them, you don't grow the bacteria to high levels, or it's very easy to deposit, which is what the selling point for the Petri film is, because you don't have to open it to look at it, you can just throw it in a set of bleach and decontaminate it. While this is true, there's still a residual risk that is there. Again, I would go back to, you have to have a really good facility to be able to do that. You can't just do that in your regular lab, because now your lab becomes a pathogen testing lab. The other challenge is that, if you really want to do it right and reliably, you also need to have a positive control, which is a pathogen that you would have to handle that. So your control strategies for these tests, you typically don't want to go through, because that now becomes much more involved to have your positive controls to make sure your test works. In addition, and this counts for the 3M Petri film as well as some other tests out there, while it is sometimes thought to be a Petri film that detects Listeria, it really detects what I would call Listeria-like organisms. What does that mean? Some of the organisms that are positive in the Petri film are not Listeria. They like it, they might behave like it, and if you find them in the same spot over time, then you probably still have to correct it. But if you send out a sample for a Listeria test, it very often might be negative. So a lot of positives in a 3M Petri film might not translate into positives when you send it out to a test lab. So it is one strategy, but I probably would continue. I would rather take better samples, smarter samples, and send them out than setting up an in-house testing lab, unless you're really a fairly sophisticated operation. Next question is, should a hold be placed on all product that is produced on a zone 1 after tested until results come back? Very loaded question, and I'm going to try to answer it to the best of my knowledge. So, what we're talking about there is, if I sample zone 1, so food contact surface, and should I ship the product that was produced using that zone 1 surface that day, or do I need to hold it in my facility until I get the result back? Number one, it depends on the test. If you test for Listeria monocytogenes, you must hold the product, because if the food contact surface is positive for pathogen, Listeria monocytogenes, Salmonella, you must assume that the product is contaminated, and you must either rework it, destroy it, or do a very, very stringent test to determine whether your product is negative. So that typically would be sampling, taking 60 samples. So that's if you test for L. mono or Salmonella, must hold. If you test for Listeria species, which is not a pathogen, In a USDA regulated facility, USDA is very clear, you can ship the product if the food contact surface is Listeria species positive. You are not required to determine whether this Listeria is Listeria monocytogenes. You're not required to recall. You have to take action, though, in terms of resampling, root cause analysis, and react very stringently. And typically, you would hold product for your second sample. So, sample zone 1, you ship. It's positive, you resample. You hold that resample. That's what USDA has always done. FDA, the expectation until this new January guideline was released was if you have Listeria on a food contact surface, you need to determine if it's Listeria monocytogenes. If it is Listeria monocytogenes, you assume that the food is contaminated. So therefore, as an FDA facility, you would hold product even if you do Listeria species testing. The new guidance from FDA aligns FDA with USDA. Would I today, if I have an FDA-regulated facility, do a Listeria species test and release my product? Probably not, because it's a draft guidance. The inspectors and in the field staff are not necessarily on the same page. If they see a Listeria species on a food contact surface, they might write you up for not determining whether it's Listeria monocytogenes. If you do this, I would, at the very least, initially have a conversation with my inspector to understand whether they're already following the draft guidance or whether they're waiting for a final guidance document. All right. Any other questions? Again, I'll repeat to everyone, we will post the slides on a web page and also post the webinar so people can either re-listen to it, which I'm not sure why anyone would do that, or you can give it to people in your company, etc., to listen to. Another question was, what do you think of Listex? So Listex is a preparation of phages that kill Listeria monocytogenes. So phages are viruses that infect bacteria. And we've seen a lot of, I'm going to say, hype about using phages to control Listeria because it's a way to kill Listeria. It is approved for use on foods in the US. It is also used by some companies, at least try to control Listeria in the environment. We have done some work on Listeria phages. I'm not saying Listex specifically, but on Listeria phages, and have found that actually some of those Listeria that survive in processing plants for 10 to 12 years are resistant to phages. So the Listex would not necessarily kill the Listeria. We've also shown that Listeria can become resistant to phages very quickly. So that means even if my Listeria is killed by the phage, over a matter of days or weeks, it might become resistant, but it is not killed anymore. My recommendation would be to focus on sanitation, environmental control, sanitary equipment design. I think the money is better invested there than on the Listeria phages, particularly in the environment. It can, under very specific circumstances, become part of the toolbox that allows you to reduce growth of Listeria on a food product, but would not be my go-to for routine control of Listeria monocytogenes on products. In general, the problem with Listeria control is it's hard work, requires constant vigilance, as I think said in some Harry Potter movies, I'm not sure, and is not an easy thing. Anything that sort of promises a quick, easy fix tends to probably not be as good as we think it is. So specific situation, when carefully think through it, it might be something, but a sort of a general practice that everyone should use and that you should expect to be effective under all conditions, probably not. Okay, since we're not getting any more questions, I want to thank everyone for dialing in. We'll follow up with some emails that will indicate to you where you can access this. If you have any other questions, feel free to email me. I'm going to type my email address into this box here, which everyone should receive. If not, a search for my name and Cornell University will get you to my email address, and we'll try to address them, including if you're interested in either other training opportunities or maybe some help setting up a validation program. Without any further questions, that is it. Again, thank you for dialing in, and have a good rest of the day. Bye-bye.